veoma čast ugostiti kolegu, profesora na sveučilištu u Los Angelesu, M Slavicha. As you know, by his surname, and also from a short beta that he sent, George has Croatian roots. He has a large family in Croatia. He loves coming to Croatia. He even took a 5-year fellowship at the University of Zurich to be able to come to Croatia more often, besides that he is doing research there. We have met 5 years ago, I think, when Philip Zibarda was visiting with us, and George came over with him and was also one of Phil's hosts in Croatia. He showed him some of the most beautiful parts of Croatia, including the most beautiful island, which is called Brač, of course. I'm not going to talk about George's career, nor his research, because he's going to do that. But let me just finish by saying that we are very happy that you come and on your travels, stop by in Zagrebu and give us this lecture. We all look forward to that.
Thank you. Well, I can say that it's just a real honor to present here, because here I feel like I'm in my heritage home, and you have so many cameras here that I feel like I never left Los Angeles. So you see, I can go anywhere and I have the best of both worlds. It's a real pleasure to speak with you, and I thank you in advance for coming at 5 o'clock when we should all be eating. But they've assured me that at 5:30 they're bringing dinner for all of us. Well, anyway, I'm trained as a clinical psychologist, but my primary interests are in life stress and how stress affects our psychological health and our physical health. And as you probably know, psychology was mostly all behavioral for its entire history, and now what we've seen is the emergence of so many amazing technologies: neuroscience, genomics, psychophysiology, genetics. And psychologists are great at acquiring these different methods and using them to their advantage. So what I want to present on today is a line of work that I'm very interested in, where we try to use these different methods to understand how it is that stress affects us neurobiologically, and how those effects influence our body and ultimately create disease. Does that sound good? If it doesn't, then you just leave. Anytime. So let's get started. Why care about stress? Well, in the United States, we are not doing so well. This is a report that was just published by the Institute of Medicine. Life expectancy, they compared the life expectancy in the United States to 17 total countries, 16 other comparable nations. And the life expectancy for men is, we rank last. The life expectancy for women in the United States is second to last, and Croatia is much further down here. So we have the shortest life. Congratulations. But we congratulate ourselves too, because we're in last place. And women, by the way, were in the middle of this distribution just 10 years ago. And although women have increased in life expectancy in most countries, they're now second to last in the United States. Well, if you look at quality of health, then you see that the number of people who die from chronic diseases is greatest in the United States, and the only country that's worse in these economically comparable nations is Denmark. Well, the United States has is the leader in infant mortality, the leader in adolescent obesity, and the leader in heart disease in adulthood. So you can see we're first place in everything, just how we like it. And it's not just that, it's that, of course, many developing nations are under high levels of stress. And we might ask the question, you know, how does stress create these chronic diseases of aging, and how does it compare to some other risk factors that we all know play a really important role in disease as well? And you might be surprised to know that in this recent meta-analytic review, they compared the effects of these other health behaviors on disease-related mortality for many of these health behaviors that we all know impact health quite strongly. Physical inactivity, drinking more than 6 drinks per day, smoking. And the effect size for experiencing high levels of stress on disease is actually higher than these other very well-established risk factors. So of course then what I'm interested in is how it can be that experiences of social stress can actually lead to disease and shorten our lifespan. So one of the most interesting things to me about stress is that stress doesn't just cause anxiety. Stress doesn't just contribute to the onset of depression. But stress contributes to a wide range of disorders, either to the onset of those disorders or to the progression of those disorders. And these disorders are psychiatry, anxiety, PTSD, Alzheimer's disease, breast cancer, ovarian cancer, obesity, diabetes, and heart disease. Worldwide, out of every 10 deaths every year, 7 are caused by stress-related chronic diseases. And of course, what's most striking about that is many of these chronic diseases are things that we could actually prevent. They're not things that are inevitable. So one thing you're probably asking already is how can it be that stress would contribute to the onset or progression of so many different disorders? I mean, it seems kind of crazy. We could make a story for how stress would cause depression, maybe because you lose somebody and you feel sad about it. But how could it be that stress could contribute to neurobiological degeneration or diabetes and heart disease? Well, that's what I want to spend the rest of the time talking about. And let me just say that one of the explanations that we have is that there might be some common biological mechanism that links experiences of stress with all of these. So the million-dollar question is, what kind of common mechanism could account for the relation between stress and these different disorders? And now, in the past decade, we have one clue to one biological mechanism that might link stress and these different disorders, and that's inflammation. So you might know that inflammation is one component of the immune system, and we usually think of inflammation as the body's response to being physically injured. So if you've ever cut yourself on the arm, hopefully not intentionally, but if you've ever cut yourself or if you've gotten poked, then you notice that you have a little bit of redness, you have a little bit of heat, and a little bit of swelling. So that's what inflammation is. But that's localized inflammation. Now you can also have inflammation throughout your body. And actually, has anybody been ill, sick in the past year, come down with the flu? Yeah. So when you come down with the flu, you feel really tired, you have a fever. Do you feel like talking to people or hanging out with people? No, you don't want to do any of those things, right? All of those symptoms of the flu are symptoms of inflammation, systemic inflammation. So localized inflammation is when you're cut, you have that redness, heat, etc. Systemic inflammation is a system-wide response that tries to fend off bacterial or viral infections. And so all of the symptoms that you have when you're sick, fever, which is to limit the spread of infection, this pain and swelling and heat localized, all of those are to kill off the bacteria that have been introduced in the body. Now what's amazing that we found out? Science published this article 3 years ago now in 2010, and they said that inflammation is one of the most important discoveries of the past decade. Why? Because they realized, we have realized now, the scientific community, that inflammation not only increases when your body is physically damaged, but it also increases when you're exposed to stress. Got that? It's very interesting. That means that your body has the same biological response to physical threat as it does to social threat. Now, I said that I started by talking a little bit about psychology and what's happened over the past decade. This is what psychology looked like 10 years ago. You had loneliness or social isolation, rejection, social evaluation, and we had some idea of the biological system that was involved in promoting disease, but we had no idea what was happening between the experience of stress and disease. Well, now, as I said, what psychology has done is incorporated so many different new technologies, tools, and methods to be able to image, right? We use neuroscience now to look at the brain systems that are engaged when you are exposed to social stress, like being broken up with or having an argument with a partner. Then we can use psychophysiology measures to look at how the hypothalamic pituitary adrenal axis, right, the HPA axis, or the sympathetic nervous system responds to stress. We can also use genetic and genomic techniques to look at how the cells in our body change as a function of these physiological systems. And we can use other strategies from immunology to look at how components of the immune system change in response to stress, and look at the relevance of those changes in the immune system for these diseases that I talked about. So even 10 years ago, we couldn't look at all of these mechanisms that occur when we experience stress, and now we're able to do all of this. And the cost of it is getting cheaper and cheaper every day. I want to switch to some data so we can talk about some of the work that my lab at UCLA has done to answer these questions about how stress affects health. And the first question I want to ask is, out of all of the different types of life events that you can experience, which ones are the worst? And if we know which ones are the worst, can we use those types of life events as a model for understanding how stress affects health? So this is one study we conducted. This was when I was an undergraduate at Stanford, and we asked the question, how are major life events associated with the onset of depression? And if stress is associated with depression, does that effect differ by the type of life event that you experience? So we brought in 100 individuals that were all diagnosed with major depressive disorder. Most of them were women, which is typical in research in general, but it's also typical in clinical research. They're recruited from this Bay Area, and they had to be depressed, but they couldn't have any other disorders. So they were depressed at the time that we assessed them. We looked at whether or not they were depressed, and we also interviewed them to examine all of the stressful life events that they experienced in the year prior to when they got depressed. Okay? So what we can do with this study design is we can look at whether or not different types of life events are differentially associated with onset of depression. This is the system we used for measuring stress. You don't have to know the details, you just know that we did a lot of work. So this is a 2-hour interview where you sit down with the participant and you go through so many different life domains. You ask them about interpersonal problems, financial problems, housing problems, whether or not they live in a good neighborhood or a bad neighborhood, whether or not they're caregiving for somebody who has a chronic disease, whether or not they weren't able to find a job for a long period of time. So all of the different stressful life events that you can think about. And then this information is then summarized and presented to another group of people who rate those stressors. And that process takes another hour, so it's very time intensive. Now, we thought, we had a hypothesis that out of all the different life events that people could experience, that probably interpersonal loss events might be some of the worst, and we know because when we lose a loved one, it seems to be associated with depression. But as you probably realize from your own lives, that not everybody loses someone who's important to them under the same circumstances, right? So some of the best examples are a self-initiated separation when you break up with your boyfriend or girlfriend. Mostly boyfriends, since there's like 3 guys in the room. Okay, so when you break up with your boyfriend, okay, you have a little bit more controllability, not a lot of social evaluative threat, and not a lot of loss of social status or regard. But when you're broken up with, okay, in that case, you're still losing somebody who you really love. But the social psychological characteristics of that experience are very different, right? Now you have much less controllability, there's a lot more social evaluation and social evaluative threat, and there's the potential for a loss of social status. So what we did was we took these types of events and we gave them a name, targeted rejection, social rejection that's directed at and meant to affect a single person that involves an active and intentional severing of relational ties with that person. So we thought that out of all interpersonal loss events, these ones that were low in controllability, so that were high in social evaluative threat, and high in loss of social status or regard would be particularly strongly associated with onset of depression. So what you see here is these are all, we limited the analyses to only life events that were very severe. Okay, so we're only including events that were severe, and then we defined those events as whether or not they involved targeted rejection or whether or not they didn't. And then we simply looked at the number of days that occurred between when they experienced that life event and when they were diagnosed with depression. And if you experienced a severe life event that involved targeted rejection, you became depressed in 1/3 as many days. So the depression occurred 3 times faster than if you experienced an event that was the same severity, but that didn't involve that type of rejection. So all of the events were severe. If these targeted rejection events occurred at work or in a relationship, it didn't matter. So what's a targeted rejection event at work?
When you're fired?
Exactly, when you're fired, versus when you're laid off with 50 people. Right? When you're laid off with 50 people, you can attribute it to other things, but when you're fired, that's a different story, right? It's very targeted. Okay. So these life events in this category involve very severe things, including deaths. Right? But deaths don't involve rejection. Okay. This is another study that we did. This is a similar type of design, but we brought people in for many more times. We asked, how are major life events associated with onset of depression? So this last study I showed you was the timing of onset. Now we're focusing on just risk for whether or not they experience a depressive episode, yes or no. Okay? This is a sample of 147 adolescents. They were at high risk for depression, but they had no personal history of depression. And by high risk, I mean that they had a mother who was depressed during their lifetime. We know that adolescent girls who have a depressed mother are among the group with the highest likelihood of becoming depressed. Almost 50% of daughters of depressed mothers go on to experience depression during their lifetime. It's a shocking statistic. So we sampled these women, we brought them in and we assessed them every 6 months for 2.5 years. At each of these time points, we gave them the same interview I told you before, where we asked them about all of the life events they experienced. And then we also diagnosed them at each of these time points to look at whether or not they were depressed. Okay? So we know the life events they experienced at each of these time points and whether or not they were depressed. And then we just asked the question, what was the risk for being depressed as a function of experiencing different types of stress? Okay, straightforward. If they experienced a severe life event that didn't involve social rejection at all, then their risk for depression increased by 40%. Okay, the odds is 1.4. If they experienced a severe life event that involved some aspect of social rejection, so somebody said that they didn't like them, or there was a short-term breakup, but the people were still together in the long run. That's what a social rejection event would look like. Then risk for depression was, the odds ratio was 6.7. If the event involved social rejection and there was and the social bond was broken, then risk for depression, the odds ratio is 12.9. And this 9.2 means that when the severe life event also involves targeted rejection, that the likelihood of becoming depressed is almost 10 times greater than if you experienced any other type of severe life stress. Okay? So now we have some clues about what types of stressful life events are particularly bad. And I have the hypothesis here that being socially rejected is among the worst things that can happen to you. Isn't this a very uplifting talk so far? Here I am, I have you at 5:30 at night, you're starving, and we're talking about being socially rejected. Okay, so we know that inflammation is involved with many of the diseases I told you about before, cardiovascular disease, some types of cancer, diabetes. We know that from research in all of these different fields, okay? Now, in order for inflammation to mediate or to link stress with disease, we also have to show what? That stress increases inflammation. Now, if we can show that, then we have the emergence of a biological model for how stress could cause disease by influencing inflammation. Now, this is a very interesting question because, as I said in the beginning, this means that the activity of your immune system is somehow influenced by all the things around you, which is a really crazy concept because, as I told you, we've all thought about the immune system as being involved in dealing with physical injuries. But what I want to say here is now to look at what evidence we might have for whether or not stress influences inflammation. Okay, we asked this question in a very simple way to start out with. This is 60 participants. They're all adult women. 30 of them were caregiving for a husband who was experiencing dementia. 30 of them were control participants. And we examined, using a computer-based system that I've developed, we examined all of the major life events and chronic difficulties that they experienced during their lifetime. And then we also examined the levels of inflammation that they were experiencing. And the way that we do this, in a very quick and simple description, is we poke a hole in their arm, we take out the blood, we put the blood into this very expensive, fancy machine. It does some things, it shows you some outputs, and then what you can do is you can get a level of these proteins that are called pro-inflammatory cytokines. So if you've taken immunology, you know that pro-inflammatory cytokines are the major drivers of systemic inflammation. Okay? So we just asked a very simple question. This is totally correlational. Do individuals who experience more life stress over their life course, do they have higher levels of inflammation? Okay. This inflammatory marker here is called interleukin-6, or if you're cool, you can say IL-6. When you're talking to your friends, so you're like, how's your IL-6, man? Okay, IL-6. Participants who experienced more cumulative life stress had higher levels of IL-6. She's totally on board. Let's go, guys. IL-6, man. All right, so why do we care? We care because people who study cardiovascular disease know that if you have levels of IL-6 that are higher than about 2.1, then you have a very high risk for developing coronary heart disease, which can lead to heart attack and death. That's why we care about inflammation. Okay, we can also do really fancy analyses. This is, I think it's very Christmas-like, so it's called a heat plot, but we'll call it a Christmas plot. What we can do with this heat plot is we can ask the question about all of the different types of stress that people experience. Which types of stress differentiate people with high levels of IL-6 and low levels of IL-6? Okay? That's the goal of the heat plot. And the way that you interpret it is you look for those cells that are bright green. Okay? So you see this is a bright green cell here, and the 5 here means that this is the highest, the most severe type of stress that they can experience. Okay? Now green here means that these types of life events, which are listed down here, these are the codes, okay? This type of life event is experienced much more often by people who have high IL-6 than people who have low IL-6. So what we want to know is what the heck is this type of, what's this stressor that differentiates people with high inflammation and low inflammation? And so I can interpret these codes by looking at what this variable name is associated with. And the question that the participants were asked was there ever a period of time when someone very close to you was being emotionally, physically, or sexually abused? So that experience early in life has a very strong effect on whether or not these participants had high levels of inflammation versus low levels of inflammation. And if you look at these other stressors that are also very green, what you find out is that all of the stressors that differentiate high and low in people with inflammation all involve elements of social stress. Either more serious social threats, like being neglected or physically abused, or things that are more common experiences like major divorces over the life course or major breakups. Okay, now this is totally correlational, right? So you're like, well, yeah, it could be that stress is causing inflammation, but it could also be that maybe somehow inflammation is causing stress. Let's say if I have high levels of inflammation, I feel fatigued all of the time, which is one symptom of inflammation. I told you that that's what causes feeling sick during the flu. Right? The reason you have inflammation when you have the flu is because your body's trying to conserve energy. So your body, what inflammation does is it tells your body that you don't like anything that you actually like. So when you're sick, you don't want to have sex, you don't want to eat, you don't want to get out of bed, you don't want to do anything. Your body's trying to save all of that metabolic energy so that it can fight off the infection. So how could inflammation cause stress? Well, if you have high levels of inflammation, you might not be able to focus at work, because one of the symptoms of high level inflammation is concentration problems. You might not want to go to work also. So we don't know from that correlation whether stress causes inflammation or inflammation causes stress. But how do you solve that in psychology? You do an experiment. So what we did in this experiment is we brought participants from UCLA into the laboratory, okay, and we made them do things that virtually everybody hates. We told them that they were going to give a talk in front of a room. So what the setup is the participant comes in, they have a microphone, they're also being videotaped, and there are three people here who they believe to be expert raters who have clipboards on the table, and they look very stern and socially rejecting and unaccepting. Not very accepting. Okay. So you can think of these as professors if you want. That's a joke. I'll never be invited back. Okay, so this is called the Trier Social Stress Test, by the way. After they get 5 minutes to prepare a speech, then 5 minutes to give the speech on why they would be a great job candidate. Okay? And after they're done with that, we ask them to do what's called a serial arithmetic task. We tell them to start at 2,945 and count backwards by sevens out loud. The experimenter is looking at them and watching the clock. Now, if they're really good at counting backwards by sevens, then we make them count backwards by 13s. And every time they mess up, we tell them to start over. So most people find either the speech task or the mental arithmetic pretty stressful. Okay, this is how we induce stress. And what we do to look at the effects of stress on inflammation is before and after they do this stressor, we put this little collection device in their mouth, and it soaks up not saliva, but another substance that's in your mouth that you didn't even know was there, called oral mucosal transudate. That's what it's called. But actually, we can use that substance, because it's a filtrate of your blood, to take out levels of these pro-inflammatory cytokines and examine your levels of inflammation using that substance. So we put this little thing in their mouth before they before they do the speech and the mental arithmetic and after. Okay? So you can look at whether or not inflammation changes as a function of experiencing this social stress. And if it didn't, I wouldn't tell you about this. So the finding is that levels of inflammation increased significantly in the laboratory in about 30 minutes when you were exposed to a social stressor of this kind. Okay? This marker of inflammation here is called tumor necrosis factor alpha. It's a major pro-inflammatory cytokine that turns on the systemic inflammatory response. This other inflammatory marker here, as you already know from years of experience, is called IL-6 man. And it also increases during exposure to the same stressor. Okay, now, you all know what these bars are here, right? These are what? Well, they're either standard deviations or standard errors. In this case, they're standard errors. But that refers to variability, right? It means that when I go through this social stressor and you go through this social stressor, that what? Our response is not going to be exactly the same. Now, that variability can occur because of lots of different reasons. But one of the things that we were wondering is whether or not individuals who might be more neurally sensitive to social rejection or social threat, we wondered whether individuals who are more neurally sensitive might have greater inflammatory reactions than people who are less sensitive. Okay? Now we know also that the brain, it plays a real critical role in appraising the types of stressors that you experience, right? That makes sense. We also know that the brain plays a really critical role in regulating the immune system. The brain is the major organ that regulates the immune system. And so as a result, differences in neural responses to social rejection might be a predictor of differences in inflammatory responses to social stress. So what we did is we brought a subset of participants back into the laboratory and we scanned their brain while they were being rejected. And now the question is, what brain regions would we care about? Well, I told you that inflammation is the body's primary response to what? Injury, right? Or physical pain, right? You have physical pain when you're injured. So one hypothesis is that brain regions that are involved in experiencing those physical threats or physical pains might also be involved in regulating inflammatory reactions to social pain or social rejection. And one of the most fascinating findings over the past decade has been that if you put people in an fMRI brain scanning machine and you expose them to physical pain by putting a heat on their leg, okay, or if you bring them into the scanner and you reject them, you make them believe that they're being rejected, then the exact same brain regions are active. Isn't that amazing? Yeah.
I'm sorry, how do you say one person that he is being rejected when he's scanned? Is that believable?
So let me answer your question and then you can tell me if I actually answered your question. So what we do is we make them believe that they're rejected. I'll show you that in a second. And then we can also ask them after that the extent to which they think they're being rejected. And then what we can look at is the correlation between neural activity in these areas and whether or not they say that they were rejected. So that gives you some indication that this brain region might be involved in the experience, their self-report of them feeling rejected. It's a very important question. But as I'm saying, this brain region might not only be involved in experiences of rejection. In fact, these brain regions have been known for a long time to be involved in experiences of physical pain. Okay, so in these brain regions, not that you have to memorize them, there's no test, but this part is called the dorsal anterior cingulate cortex, or if you're cool, you say DACC. These brain regions are called the anterior insula. If you're referring to both sides, then you say that bilateral anterior insula. What we did was we brought participants into the laboratory with 2 other people. All 3 people showed up at the same time. We put them in the brain scanner and we told them, you're going to be playing a ball tossing game, okay, with these 2 other people. So now you all go to your rooms, okay? Now, you're all participants in this study. You lay in the brain scanner and you have a button box, okay? Now, in this game, you're wearing goggles, so this is what you see when you're laying in the brain scanner, okay? Every time I receive the ball, by pushing a button, I can either choose to throw the ball to Bruce or Harold. Okay? These are the other 2 participants, and everything's going really great. Looks like Bruce and Harold like me, so they keep passing me the ball. But halfway through the experiment, guess what happens? Bruce and Harold stop passing me the ball, and now I'm laying alone in the scanner, and I can't throw the ball to either of them because I never get the ball. Okay? So this is how we induce, in a small way, an experience of social rejection. All right? Okay. So what happens? Well, what we see is that the primary site in the brain that's active when you expose people to social rejection is the DACC. This is the same part of the brain that lights up when you expose somebody to physical pain. And if you look at the amount of activity in this brain region, then you see that it's highly correlated with the extent to which people increase their levels of inflammation to the public speaking task. Okay, so these are 2 experimental sessions that happen back to back. If they were more neurally sensitive to social rejection, then they showed greater inflammatory responses to social stress. These are the first data that we know about to show that these brain regions that are involved in perceiving the world, but also responding to physical pain, are involved in increasing levels of inflammation to threats that are purely social, right? Okay. We since did this task again with a slightly different study setup. Well, you don't have to know all of these things, but in this study, the participants came in, we poked a hole in their arm so that we could take blood samples at 1, 2, 3, 4, 5, 6 different time points. In the middle of this experience, so these are the baseline time points and these are the post-stressor time points. In the middle of that experience, we did a social evaluation task, and I'll show you what that's about in a second. And the question here was, can we replicate this finding that exposure to social evaluation or social threat increases levels of inflammation in the blood? What we did in this study is that we had 1 session, we brought participants into the room, and we interviewed them about their childhood, about their family life, about what their parents did, about their goals and their hopes and their aspirations for the future. We videotaped all of that. It's kind of an intimate thing. I mean, you usually don't talk about all that, right? And then what we did is we brought them in to this second session here with another participant, and we said we said that we'll randomly assign one of you to go into the fMRI scanner, the brain scanner, and the other person is going to watch your video and judge you. And when you're laying in the brain scanner, you will see what they think about you. Okay? So we flip a coin, and then we say, okay, you're the participant, so you're the one who's going to be judged, and the other participant, you're the one who's going to go into the control room and watch the video and make the judgments. Now, of course, that person works for us. It's not actually another participant. And it's not random. Obviously, the participant is always the person who goes into the fMRI scanner. But as social psychologists, you know that we're all tricky like this. So this is the setup. The confederate is in the scanner control room. Okay, they sit at the computer. We also wrap their arms so that the participant believes that both people are actual participants. But her arm is not actually poked. She's watching the video here, supposedly, and making her ratings here, and the participant is in the brain scanner. What does the participant see? This is what the participant sees when he or she is in the brain scanner. The task here, they believe that the participant in the control room is identifying different words of how the participant is coming across in the interview. Oh, interesting. That's pretty nice, right? Okay, and insecure. Now, this is totally standardized. This is a video. Everybody believes that they're actually seeing somebody else choosing these words, but everybody sees the exact same video and the exact same sets of words. Okay? Now, this experience of social evaluation is enough to increase inflammation in your body. Inflammation starts low. This is the social evaluation, and inflammation goes up. This is interleukin 6 or IL-6. If you impose that line for heart disease risk, you see that just with this social evaluative stressor, we can get people's levels of inflammation up above levels that increase risk for heart disease. Now, I'm not saying that something like this increases risk for heart disease, because people go right back down, right? Pretty quickly in about an hour. But it means that if you had chronic experiences of social stress or social threat like this, that you can get people's inflammation levels up high enough that over time it can increase risk for heart disease. Now, if we look to the brain and we say, can we identify areas in the brain that are associated with risk, with increases in inflammation? We can. In this study, the primary site of activation that's highly correlated with levels of inflammation is the left amygdala, and you probably know that the amygdala is very involved in responding to emotional threats and fear responding. So people who were more neurally sensitive to social evaluation showed greater inflammatory responses to this task. Okay, so I've told you that adverse social experiences like social evaluation and social rejection can reach inside the brain, and that these stressors can influence the periphery to increase inflammation. But how far do these effects go? I mean, social rejection couldn't influence the activity of our genes, right? Right? Okay. You ever checked out a cell, an immune cell lately? Okay, this is what your immune cells look like. They're not quite as pretty. But you have a lot of these immune cells floating around. All of you do, I guarantee you. What do you have to know? You have to know that this is the wall. This is the cell wall. Okay. For those of you who are not immunologists or biologists, your immune cells have these receptors that are on the cell wall. Okay. And these receptors can listen for a lot of different things. Okay. One thing that immune cells listen to very carefully are signals that come from your stress axes, your physiological stress responses. And primarily, they listen to two end products of the fight or flight systems, cortisol and epinephrine or norepinephrine. Okay, these are the end products of the stress axes. Now, receptors on the walls of immune cells listen to these things. And then you have these things on the inside of your cells, in the cytoplasm of the cells, called transcription factors. Okay? Now, these transcription factors, they go up and they say, "What's up, receptors? How you doing? You know, give me your message." Okay? Then they take that message, and when it's the case that these two transcription factors let go of this transcription factor, okay, they stop holding hands, then this transcription factor can walk on over to the inside of the nucleus of the cell. They can turn the gene on, and when the gene is turned on, then the cell produces something. And since these are immune cells, what these immune cells produce are pro-inflammatory cytokines like tumor necrosis factor alpha and interleukin 6. Okay? So you're all watching, this is the schematic. So you have a stress signal that gets picked up by the receptors. These transcription factors let go of NF-kappa B. NF-kappa B heads into the gene in the nucleus. It leads to increases in mRNA, and mRNA then get translated into proteins, in this case, pro-inflammatory cytokines. So what we can do experimentally is take blood from participants. We can go inside the cells, and we can measure levels of these intracellular transcription factors to see whether or not certain types of stress have the ability to go inside the cells and turn on the genes. Okay, see what we're doing here? Okay, before I showed you this schematic. It's the study that we conducted where we had participants interviewed at every time point over 2.5 years. Remember I told you that we interviewed them to ask them about the life events that they experienced before each of these sessions, right? And we also looked at whether or not they were depressed. What I didn't tell you is that we also took blood samples at each of those sessions, okay? And we can take out that blood, we can put it in the big fancy machine, and we can use that machine to look at levels of intracellular transcription factors to identify whether or not the genes are turned on as a function of experiencing some type of life event. And this is what we see. These are all events that were severe life events, okay? And those severe life events either involved targeted rejection or they didn't, okay? If the severe life event that they experienced involved targeted rejection, they had higher levels of this levels of mRNA for for NF-kappa B. They had higher levels of NF-kappa B at visits when they had recently experienced a targeted rejection life event compared to visits when they didn't. Okay, so it's all within subjects. And you see the same thing for this other marker of gene expression, inhibitor of kappa B. They had higher levels of inhibitor of kappa B at visits when they had just experienced a targeted rejection life event compared to visits when they had it. So experiencing rejection can not only cause negative thoughts, not only influence levels of inflammation in the body, but those signals have the ability to go inside your immune cells at the molecular level and influence the activity of the gene to actually turn genes on. Okay. Now, you might be wondering, if you haven't already figured it out, why on earth would our body do this? Right? You're thinking like, now you're imagining like all the exams that you had and you're like, my body is so stupid. Like, I'm in exams all of the time, why, you know, or you have job stress all the time, why is my body doing this? It sounds counterproductive, right? It doesn't, I haven't told you anything good about it yet. Right? Well, we don't know why for sure. So this is just theory, this is just speculation. But what I can tell you is that the primary job of the innate immune system is to protect the body from physical threats. I told you that. Physical threats in our historical environments involved things like predatory animals and dangerous conspecifics, people in our group, social group. And now an inflammatory response to these types of physical threats is adaptive. But what's even more adaptive is identifying those physical threats before they're even a physical threat. And the way that you do that is by identifying social threats, like being excluded or being in the presence of somebody else that seems evaluative or hostile. Okay? And what happens if you respond, if you have an anticipatory response in advance of actually being injured, then your body is actually much better able to deal with physical injury when it occurs. So one of my favorite studies was done at Stanford on this topic, and they brought in men who were all going in for knee surgery. Okay? Half of the men, they assigned them and they stressed them out before they went in for knee surgery. They had them do that public speaking task that I told you about before, and then they went in for knee surgery. And the other men just went in for knee surgery. Okay? And then they just asked a simple question. Is there a difference in the recovery time? How fast the wound heals on the knee? The men who were stressed out prior to going in for knee surgery recovered faster and had better clinical outcomes. Why? Because experiencing social stress activated the innate immune system, leading to the release of pro-inflammatory cytokines throughout the body. And then when the body was physically injured, those cytokines were going to work immediately to promote wound healing and to promote recovery. So this is why it makes sense that your immune system cares about social threats, because to the extent to which social threat has possible implications for being wounded, the earlier your body detects it, the faster it recovers. Okay, let me just make one more point about this, which is that this has a lot of short-term benefits. Okay? If you're being chased down by a tiger, like that happens, if you're being chased, or if you're exposed to physical threats once in a while, then you better mount this inflammatory response, otherwise you're in big trouble. But what's the problem? What's the difference between us and animals? When a zebra is out in the wild and it sees a dangerous threat, it mounts a fight or flight response, it increases levels of inflammation, and when that threat is gone, what happens? The zebra is back to doing its business. It never thinks about that threat again. But what do we do as humans? Right? We are so darn smart that we're able to think about these threats all of the time, regardless of whether they're actually in front of us or not. So, you're also smart that you can think about that difficult job interview for 5 days before you're ever in the job interview. So what I'm saying is that, given that we're highly intelligent and we have great memory, we're able to create symbolic threats in our mind that have the ability to activate this system in the absence of actual threat. And in fact, we can mount this inflammatory response to things that may never actually happen or that will never actually occur. And it's under those conditions that this system becomes very problematic for health. Because why? You have high levels of inflammation, but no adaptive value. There's no actual threat. Okay, now we've called this phenomenon human social genomics. It's called human social genomics because for a long time we've known that social environmental experiences can influence the gene expression of animals. But this last study that I showed you is very new. And really only now, very recently, do we have evidence that in humans, experiencing social stress can influence the activity of our genome. And not just a couple of genes, but hundreds of genes. So, other studies that I didn't tell you about looked at individuals who say that they're lonely, okay, compared to individuals who say that they're not lonely. And if you just do a, very simple analysis, and you compare the genomes of individuals who are lonely versus those who aren't lonely, you see this enormous difference in the number and the types of genes that are turned on in the lonely people versus the non-lonely people. And the genes that are turned on in the lonely people are lots of genes that are involved in inflammation and in the innate immune system. Why? Because being alone poses risk for physical danger, historically. Okay? Now, you might be surprised to find out that your genes are changing the activity level all of the time. Why? Because I can ask you, are you feeling, I can ask you if you have negative thoughts, right? And you can say, no, I feel good today, right? But if I ask you, how's your genome doing today? You can't say, oh, my genes are turned on and my genes are turned off. Right? You don't have any way to access that level in your body. And so because of that, we go around the world assuming and feeling as though we're pretty biologically stable. I mean, our stomach is upset once in a while and this and that. But we think that our genome is basically stable. Well, what I'm telling you is that despite the fact that you experience your body as being biologically stable, that it's actually the genome is always changing in its activity as a function of the physical and social environments that you find yourself in every moment of the day. Now, what's most fascinating about that is that every day cells and proteins are dying in your body. And so in order to compensate for this natural process, your body actually has to replace these dead cells, has to rejuvenate those cells that have died. And in fact, 1% to 2% of your entire molecular makeup must be replaced every day. So as a result, the experiences that you have today will actually influence your entire molecular makeup for weeks and months into the future. So I'd like to say that this is a paper that we wrote on the topic. I'll show you the web address at the end, but if you're interested in this idea that stress can influence the activity of your genome, then we've summarized some of these ideas in this paper. And in the last minute, let me just say what the take-home points are. So I started by telling you that experiences that involve social threats, I mean that sort of in a general way, or social rejection more specifically, those experiences have a strong impact on physical and mental health. Now, I showed you that both naturalistically, we interviewed people about life events they experienced, and we also put them in the laboratory and exposed them to social stress. And we see that social stress, both naturalistic life events and laboratory-based stressors, can increase levels of these pro-inflammatory cytokines. It's also the case that if you do neuroscience, neuroimaging studies, the same regions of the brain that are involved in responding to physical pain also respond to social pain. And in fact, if you do these neuroimaging studies and you give people Tylenol, you all take Tylenol for pain. Aspirin.
It's the same thing.
Yeah. So if you do this neuroimaging study where you're rejecting the participant and you have them take aspirin every day for 3 weeks before they come in, they have no neural response to social rejection. So by reducing physical pain signaling in the brain, you can actually alter experiences of social pain. Okay. And then last I said, social isolation and rejection are associated with genome-wide transcriptional shifts toward a more pro-inflammatory phenotype over time. 1% to 2% of your entire molecular makeup is regenerated every day. So the things that you experience today live on inside your immune system and your genome for days and months into the future, which is what I've said here. The experiences that we've had can impact your entire molecular makeup for days and months into the future, and so I suggest that you plan your day. Okay, I'll thank you. This is the website. This is our website. You can go to the website, the papers and everything else that I discussed. If you want to see all the beautiful people who work in my group and who are responsible for helping out on these projects, they're there as well. If you have some ideas and you want to do a cool project, then my email's here, and you can email me and say, I love IL-6, and I want to do a study on it. And I'll say, I love IL-6 too, and we should totally talk. Okay, and I'll thank you, and I've been told that we have some time for questions. Thank you.
Thank you, George, for inspiring talk. We'll keep changing our genome every 30 days, which is good. Okay, so we have time for questions, applications, your ideas.
So I'll say, just as a preamble, that I avoided biology for my entire life until like 3 years ago. So there is no question that is too stupid or too basic to ask.
Just one practical question. Do
Yeah.
you really have more than one scanner, or you like your subjects?
So I realized after I told you about that I didn't tell you that for that study that people arrived. You see how much people will believe when you tell them. So we told them that they arrived. They did arrive with 2 other people, and that each of them were going into separate fMRI scanners. Of course, you might know fMRI scanners are several $1 million each, and no school has 3 fMRI scanners. But under, huh? MIT, fancy. They arrived with 2 other research assistants.
That's what I thought, but I just wanted to.
They go in there, and as soon as the participant is in the scanner, those research assistants go to class. And the ball tossing game that you saw is all controlled by an algorithm. There are not 2 other people. We're really nasty. By the way, you can also do purely behavioral research with this task. You don't have to do it in the scanner. You can bring participants into the laboratory, and you sit them down in a computer, and you say, then you take the other participants out. Maybe they work for you, and you say you're, participating in this computer game on the computer network. You can measure whatever outcomes you want. It doesn't have to be neural activity. Yeah, please.
I have a question regarding general anxiety disorder.
Yes.
Because I think that once I read something about that people who have that disorder would actually be pretty fit historically, because they would anticipate all the dangers and that could endanger them. So could we connect that to your research in some way? Do you have anything to say about general anxiety disorder?
It's a really smart question. So there is a lot of research looking at the association between depression and physical health problems. Specifically, chronic pain problems, heart disease, arthritis. And there is very little research looking at anxiety disorders and physical health problems. And as it turns out, it's probably the case that severe anxiety disorders are more strongly related to physical health problems even than depression. And there is not so much research yet, but what we've found is that if you look at levels of inflammation in people who are depressed, it might even be the case that the anxiety part of being depressed could be what's driving the inflammation. As you probably know, most people who are depressed also have symptoms of anxiety. And so to your point, we might end up finding out that even in the context of depression, it might be the anxiety component or the social threat anxiety component that is driving the immune system. It's a very interesting question. Yeah.
Did you maybe investigate some protective resilient factors that can lessen a level of social stress?
Yes, so for those of you who have taken a clinical psychology class, if you have somebody who has lots of negative thoughts, what's one way that you can intervene? Can anybody in this room intervene?
Have a cigarette and go play yourself.
Okay, you can drink some rakia and you can be off with it. Or you can smoke, you can do whatever. Okay, let me help you out. So if you're a psychologist and not a drug dealer, and somebody comes to you and they say, I'm under a lot of social stress, you can do cognitive behavioral therapy. Okay. You can examine the negative thoughts that they're experiencing, and you can work with them to reshape those thoughts or to restructure them or to rewrite them, however you want to think about it. Okay, that's one thing you can do. So you can change the types of thoughts they have. You can work with them to do that. You can also bring them in and have them do mindfulness. Okay, so you probably know somebody who does this mindfulness meditation where you sit and you center yourself, and when you find your thoughts floating away, you gently bring your thoughts back to the present. Okay, that's the general concept of mindfulness. So, one thing about these social threats is that you're usually thinking about things in the future, right? So what does mindfulness meditation does is it takes those thoughts that are in the future and you bring them back. And you bring them back in a non-judgmental way. So you don't say to yourself, damn it, George, stop thinking about the future. What you do is you just notice the fact that you're thinking about the future and you bring your thoughts back to the present. So that's another thing you can do. And you can also do other types of restorative, meditative techniques like yoga. Okay, that's a third method of stress reduction. And I didn't show you the data here, but there are now 3 studies that one uses cognitive behavioral therapy, one uses mindfulness meditation, and one uses yoga. And you can take people who are under high levels of stress, you can put them through that treatment regimen, and you can reduce their levels of inflammation at the genomic level. You can change back their gene expression so that it looks more like people who aren't under those high levels of stress. So one example is individuals who are just diagnosed with cancer. You have them do cognitive behavioral therapy that focuses on the negative thoughts about their prognosis, and you can actually inoculate them from some of the effects of that stress on their genome by using cognitive behavioral therapy. Yeah.
You just triggered some thoughts because I work in hospital with people who are diagnosed with cancer.
Yes.
And what was interesting for me is that what I tell them every day is basically the basis of my work is that depression can complicate, of course, and negative thoughts. It doesn't have to be cancer, it can be just something very chronic, asthma, COPD, whatever.
Yes.
But what was interesting what you said about knee injury and inflammation, because in that sense I think it helps them. So I just wanted you to comment, taking that into consideration with the knee injury and basically making them feel good or recover better if they had inflammation.
Yes.
How do you comment on that chronic illness? Does that not behave like physical injury, or the inflammation process is different, or is it behavioral variable? The depression is making the possibility less.
Yeah, it's a really great question, and there's, as you might know, there's not a lot of good work looking at the effects of stress on clinical functioning in cancer. There's a lot of clinical oncologists who study lots of really important things about cancer, but there's not a lot of great psychologists who work on these types of questions in cancer populations. So if you're looking for some really interesting topics that are really important to study, this is one of them. What we find out, did you say breast cancer?
No,
What?
lung.
Lung cancer? Yes,
Lung cancer.
lung cancer. You didn't say though, right?
No.
Okay. Okay, so you actually, I was just primed because you're wearing pink, so. That's the breast cancer.
Yeah.
Okay, so, not because it's female. Okay, so in breast cancer, for example, because I don't know much about lung cancer. So in breast cancer, for example, about 45% of women who are diagnosed with breast cancer have levels of fatigue, okay, tiredness and fatigue, that completely take them out of their routines. They can't focus on work, they don't want to socialize, they feel tired and heavy and fatigued all day long. And actually, for these women with breast cancer, that's their primary clinical complaint. That's the worst part about the treatment. For the other 50 to 55%, they still have to go through, all of the cancer treatment, but they don't experience these high levels of fatigue. And so the question is why do some of these women with cancer have high levels of fatigue and others don't? And it seems as though inflammation is a primary driver of fatigue in those breast cancer patients who have high levels of fatigue. And we just published a study that looked at the relationship between stress and fatigue. And we showed that sure enough, it's these women who have higher levels of stress over the life course that have much higher levels of fatigue. So one possibility in this cancer model, breast cancer, is that stress over the life course contributes to high levels of inflammation throughout the body, and high levels of inflammation contribute to fatigue. Now, if you're talking about other cancer models, levels of inflammation actually increase in the tumor microenvironment. That's a fancy word for like the tissues and all the things that are right around the tumor. So your body is trying to kill off the tumor, trying to attack the tumor as a foreign agent by increasing levels of inflammation in that localized area. So that's a natural thing that I think is typical for most cancers. But not all of those cancer patients experience high levels of systemic inflammation. And it seems like that's what's particularly related to poor clinical outcomes in that model. Does that address?
No, it's just an interesting question. And I did plan a project like that, but it's with lung cancer. So, because I read something that it's not, it's connected to quality of life and quality of life and genomics and personality.
Yes.
That's why it was interesting. And then, of course, it's connected to negative function depression.
Yes.
Just figured it out.
Yeah. Yeah, most patients who come in for treatment for any type of cancer, not just in Croatia, but in the States, there is very little assessment of psychosocial functioning. It's usually there's not a lot of attention paid to it, even though we see now quite a bit of evidence that psychosocial functioning has really important impacts on the lives of these people.
George, I have a hypothetical question. I think you have really nicely explained the role of the inflammation in physical injuries and also the social threat and how it has the short-term benefits of the long-term maladaptive functions. And also how it changes the gene structure. But the hypothetical question is, if there is this long, at the individual level, there's a long experience of social threats, how does it not happen that it becomes adaptive not to have the inflammation? Why are the genes not learning that it is different eventually than the physical threat, injury, or whatever?
Yeah. Yeah, so how would that work? Basically people if you die from high levels of chronic inflammation, then you would think that, that adaptation, evolution should have not selected for that, right? Well, the other side of the coin is that the reason that you have high levels of inflammation is that it's selected for. In other words, you have high levels of inflammation in response to social threat, that evolved because it was those people who got attacked and who lived to see another day. So, what is this? Two sides of the same coin. I mean, as I hear your question, you're saying, if it's the case that high levels of inflammation create diseases that kill people, right? Then why would it be the case that the immune cells haven't learned to stop responding to social threat?
Which is ongoing.
Yes, which is ongoing. And, well, the only reason I can say is that because not responding to chronic social threat kills you much sooner than responding to social threat. So, I guess a good example is they do these not in social hierarchies in humans are somewhat stable, but they change over time. But in some animal kingdoms, like with monkeys, they have pretty stable social hierarchies. Okay, so this macaque monkey is here, the other one is here, and everybody knows their place in the social hierarchy. And if you're in a stable social hierarchy and you don't have an inflammatory response and you get injured, then you die. So you die much quicker without the inflammatory response, essentially. So that's my best attempt to say that, you die later on from inflammatory-related disease down the line, but you die much sooner if your hand is chopped off.
It doesn't matter if you die later on because you have already reproduced yourself. So your genes have passed on.
Thank you. Take that.
Okay.
And even if it wasn't so, evolution needs a large time span to work in a stable environment. And we live in such a crazy way we live, only for the last, let's say, 50 to 100 years, so evolution has not had a chance to do anything.
Okay, we'll. I want to thank you, George, for your presentation. It was very intriguing, and for many of us, very new. So thank you. And I just want to make a disclaimer. His visit here was not sponsored by Tylenol nor aspirin.